Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. How are you, dear students? I hope you all are fit, well, and enjoying your online studies at your home. Dear students, I welcome you in Pakistan International School Types Virtual Learning System for the session 2020. And this is biology class for grade first year. Dear students, in today's lecture number 31, we will study about cellular respiration. We have started this topic, cellular respiration, in chapter number 4, bioenergetics. Today we are gonna see its next topic, that is electron transport chain, that is also called ETC, and we will have chemiosmosis, in which ATP production is occurred in every cell of the body. So let's start. Let's take a review of cellular respiration, which we have studied up to now. That in a first step which we have studied, that was glycolysis. If you see, this is glycolysis, in which glucose is broken down into pyruvate. 6-carbon compound glucose is broken down into 2 molecules of 3-carbon compound, that is called pyruvic acid or pyruvate. We have studied in detail, that is glycolysis. In glycolysis, just a little amount of energy is produced in the form of ATP, but that energy which is stored there, that is in the form of NADH. It means electrons are carried by coenzyme NADH. Now this nicotinamide adenine dinucleotide hydride, now it will move to the further reaction in ATC. Then after the glycolysis, we have studied Krebs cycle. When pyruvate enters into the Krebs cycle, it will change into acetyl coenzyme A, and then it will move into the Krebs cycle, in which carbon dioxide is released, as well as just a little amount of ATP is released. More of the energy which is consumed by electron carrier, or electrons are carried, or that energy is stored in the form of electrons which are carried by NADH and FADH2. Now these NADH and FADH2 move to electron transport chain, or that is called ETC, and then they change into ATP in chemiosmosis. Okay, so we will study now this ETC and chemiosmosis, when the more energy in the form of ATP is produced. So you can see here ATP summary, like in glycolysis we get 2 ATPs, Krebs cycle we get 2 ATPs, and ETC we get almost 32 to 34 ATPs. So what is electron transport chain? What is actually is the reaction? So electron transport chain actually is the series of complexes that transfer the electrons from electron donor to electron acceptor via redox reaction, and couples this electron transfer with the transfer of protons, H plus ions across a membrane. So this proton gradient, that is also termed as electron, that is also termed as electrochemical gradient, and that is actually the force which drives the synthesis of ATP, that is adenosine triphosphate. And the molecules of chain include peptides, enzymes, and some other protein molecules. And this is happen only in aerobic respiration. This doesn't occur in anaerobic respiration, because in this reaction, in the last Oxygen is utilized in the form of a final or terminal electron acceptor. So now we will see electron transport in detail. So to see this one, first you see the components of electron transport chain. Components of electron transport chain is having almost 5 complexes. The fifth complex is for that is we can say ATP synthase, or that is utilized for chemiosmosis. The fourth, number 1, 2, 3 and 4, they are utilized for the electron carriers. So in complex 1, we are the complex is said to be NADH dehydrogenase, and actually this is the electron pump by which, sorry, proton pumps by which protons move inward into out of the inner membrane, like into the matrix of mitochondria. So in complex one, there are some proteins which are embedded, that is FMN and FES. FMN is called flavin mononucleotide protein, and then we are having FES, that is termed as iron sulfur complex. In complex 2 actually is for specifically FADH2 molecule, of which receives electrons from FADH2. So that is also called succinate dehydrogenase, because we have studied previously in Krebs cycle where the succinate is formed, FADH2 is formed there. So FADH2 directly from succinate, it moves to this compound, this complex, to FAD, where FAD and FES are, we can say, the electron carriers. Then we are having complex 3, ubiquinone cytochrome C reductase, that also called as the proton pumps, and it is having heme group inside, and FES iron sulfur complex also is present in this compound, in this complex, and this complex number 3. So there is the now series of cytochrome compounds, cytochrome proteins. Like some are embedded in complex 3 and some are embedded in complex 4, and intermediate cytochrome is also, that is called mobile electron carrier, is also present there. So complex 4, this one, that is having cytochrome oxidase, that is called as cytochrome oxidase complex 4, that is also proton pump, and that is having a heme group in which FES is present, and then copper, cuprous A and cuprous B are present. So that we can say the cytochrome A and cytochrome A3 are present here. Now we will see cytochrome C. Cytochrome C, you can see this, the intermediate protein, that molecule, that is the mobile molecule, which transfers electron, takes from complex 3 and just give to the complex 4. And then we are also having ubiquinone, non-protein component is this, and just it is the mobile carrier which carries electron from complex A, and it provides through complex 3. Complex 2, as I have mentioned, that is succinate dehydrogenase, that is the intermediate, like, or that is the complex for FADH2 specifically, and here it also gives electrons to ubiquinone, and then ubiquinone just transfers these electrons to complex 3. Now we will see the steps of electron transport chain and chemiosmosis. First of all, what happened? The molecules from NADH2, if you see this one, gives two electrons to NADH dehydrogenase. We have seen Krebs cycle, that you can see from Krebs cycle here at this place and at this place NADH are formed. So this NADH gives the electrons to the first complex, and here it donates electrons as well as protons. When it donates electrons and protons, so it just oxidize into NAD, and then what happened, this NAD again just moved to the Krebs cycle for the carrying of the next electrons and protons. Electrons pass through the ETC energy, releasing energy, as we have mentioned in photosynthesis. When electrons move from one carrier to another carrier, it reduces its energy, and this releasing of energy is used to pump out the protons from inner of the like this is matrix of Christie, that is in the mitochondrial inner membrane, and then it pumps the hydrogen or protons to outside of this membrane but inside the mitochondria. Buildup of proton ions in the space creates an electrochemical gradient when protons from NAD, and this is succinate, will change into fumarate by complex 2. From here FADH2, it moves directly to succinate dehydrogenase, and then proton and electrons are removed from them. Protons move to the next of the membrane, and electrons move to the carriers and then into the complex 3. By this movement of protons from inner of the membrane to the outer of the membrane, out of the membrane, electron, or that is called electrochemical gradient or proton gradient, is formed. So these proton ions want to diffuse back across the membrane, but that is unable to do this one. So they use ATP synthase or ATPase complex, that is facilitated diffusion is done by the utilizing of this complex, and proton moves inside. By using this proton and energy is released from that, that energy is utilized to phosphorylize ADP into ATP, that is highly energized molecule, which actually we want from this at the end of this reaction. So you can see here, okay, now we will see here in the process of ETC. First of all, what we can say, that is inner membrane space, that is mitochondrial matrix. So this is upside down, which we have seen before, and this is the membrane, inner membrane. Mitochondria, if we can see, this is mitochondria. It is double membrane, outer one is smooth and inner one is having this type of zigzag structure. So actually this membrane is like this, sorry, it is like this, and if we see here on this membrane, these electron carriers are present. So like this, on whole of the membrane electron carriers are present. So the electrons and protons now move from inner of the membrane towards out of the membrane, but that is the matrix, and but they remains inside mitochondria. So first reaction which we see here, that is a redox of NADH plus H at complex 1. It means redox reaction occurs. The reduced NADH will change or oxidize into NAD, and electrons go to complex 1. 4 protons pumped out of the out of complex 1 in response of the oxidation of NADH. Number 2 is redox of FADH2, like that is on complex number complex 2, which is embedded here, and that is also the coenzyme Q. Here the coenzyme Q picks up the electrons from complex 1 and complex 2 also, and transport to complex 3. Redox of complex 3, here 4 protons pumped out of the matrix from the matrix to intermembrane space, from the matrix to the intermembrane space, and carriers C transports electrons to complex 4. That is C is the cytochrome C that transports the electrons from complex 3 to complex 4. Then redox of complex 4, you can see 2 protons pumped from a matrix to intermediate, from here to the outside, from the intermediate intermembrane space, and formation of water is done, and 20% of water is formed by this process, which is present in the body. So look, a pair of electrons are coming down. So these electrons have to be accepted by some other compound or molecules to just produce more energy, and to to reduce the wastage of the energy. So what happened? Oxygen which we inhale, now it is utilized here. That's why it is called aerobic respiration. So 2 protons from this matrix, what happens, that are that react with oxygen, and that change into water. So in the last, the terminal electron acceptor is water. So now ATP synthase action pumps protons intermembrane space to matrix, produces ATP. Now in intermembrane space, protons are in gradient, more gradient, so they move inward through this this complex, that's ATP synthase. And then by movement of proton, this energy is utilized to phosphorylize ADP plus PI to ATP. So here we can say that NADH plus H plus, that is oxidized to NAD plus, and also 2 protons are removed from them and 2 electrons. These electrons are carried by the complexes, and this proton make the gradient to flow from ATP synthase. And this NAD moves back to the Krebs cycle. So likewise, 2 protons from the matrix, and 2 electrons, they are accepted by molecule like oxygen atom, and we will get water, and this water which is formed in our body. So that oxygen is utilized as we can say the terminal electron acceptor, and that is also called terminal oxidation reaction. Okay, so last step of this electron transport chain is to produce ATP by ATP synthase, and that is done by we can see the complex 5. And complex 5, you can see that this is embedded in a lipid bilayer of what inner membrane of mitochondria. So that is outside of this one and inside of this one. It is composed of multi-protein enzymes, and it utilizes the electrochemical gradient of proton to form ATP, as well as that is made up of, if we see its structure, it consists of two domains, F1 unit and F2 units. F1 and F naught unit, F naught or F0, we can say this one. The enzyme required for this synthesis is called ATP synthetase, which is also called oxysome, or that is also called, sorry, stalked particles. If we see their structure, so F naught and F1 particles, so we can see here F naught particle is embedded in lipid bilayer, and it is composed of a rotor stator, like which drive the force of protons to outside of the from the matrix to the inside of the membrane. If we see its F1 particle, that is called as a head, and here ADP is phosphorylized into ATP, and it is also having alpha, beta and gamma particles, and this is called B2 complex or stator, part of the stator, which drives the force which runs or movement of this a whole like apparatus is done by this B2, and that is present inside the inner layer of we can say the mitochondria. Okay, what is the process of chemiosmosis? If we see, we have discussed before, but right now this is the last step which we'll see. Here you can see proton pump, that is complex number 4. That proton is like pumping out, and from here the electrochemical gradient is formed. So rotor, that is F0 domain, is there, so it just runs circular motion, so by which protons move inside of this particle of this now from F naught particle, and then moves to the to inside. And then what happened when proton moves outside, this energy is utilized, that is called a stalk or head. This energy is utilized to phosphorylize ADP into ATP. This is F1 domain at which ADP is get phosphorylized. So the flow of proton through F naught channel induces F1 particles to function as ATP synthase, and the energy of proton gradient is used in attaching of this phosphate, inorganic phosphate radical to ADP by high energy bond. As we know that phosphate group when attaches to ADP, that is high energy bond, when it is released, so it released almost 7.3 kilocalories per mole. And oxidation of one molecule of NADP, which we have seen before, that gets here we can have ATPs from. Then how much we will see right now. So Peter Mitchell, that received a Nobel Prize in 1978 on the discovery of chemiosmosis process and production of ATP in mitochondria and chloroplast. So if we see here all about that energy, so NADP or NADH, that produces NADH2, that produces 3 ATP in chemiosmosis process, and FADH2 that produces 2 ATPs. 2 ATPs in chemiosmosis process. So at the end of this process, we get almost 32 to 34 in electron transport chain, and then the remaining in the previous steps. So if we see the summary of energy currency of cellular respiration or energy budget of cellular respiration, so now we can see here that in glycolysis we get 2 ATPs. In glycolysis, we get 2 ATPs and 2 NADH2, but 2 ATPs are utilized in glycolysis. So we can say that we have 2 NADPH2, so it 1 NADH, it gives 3 ATPs, so we'll get 6 ATPs in glycolysis. But as we know that we have utilized 2 here, so that is minus 2, so we'll get 4 in glycolysis, 4 ATPs in glycolysis. And if we see the NADH production, so from pyruvic acid to convert into acetyl coenzyme, we get 2 more NADH. So 1 NADH produces 3 ATPs in electron transport chain, so here we'll get 6. Then in Krebs cycle we get what we can say, the 6 NADH and 6 into 3, then we will get 18 ATPs in chemiosmosis, and one FADH2 changes into or form 2 ATPs. So they 2 FADH2 produces 4 ATPs. 2 FADH2, we get 2 molecules of FADH2 in Krebs cycle. So 2 ATPs in the form of GTP we form in a Krebs cycle separately. So overall, human eukaryotic cell that forms 36 ATPs. 2 in glycolysis separately, and 4 from NADH, and then again from pyruvate to we can say from pyruvate to acetyl coenzyme, it also will get 2 NADH, so we'll get 6 ATPs. Here 2 ATPs are utilized in glycolysis repeatedly. Then from Krebs cycle we get 6 NADH2, so it means we get 18 of ATPs. Then we will get from 2 FADH2 4 ATPs, and then we get 2 separate, like from a Krebs cycle we get 2 ATPs. So overall we get 36 ATPs after the end of cellular respiration. So dear students, that's it for today. I hope you understood the lecture, and inshallah we will continue our lecture and continue our topic, in which we will see substrate level phosphorylation, because right now we have discussed about oxidative phosphorylation. Then we'll see substrate level phosphorylation and some other processes of cellular respiration. I hope you understood it. Wish you all the best. See you then. Allah Hafiz.